Hello, everybody. I wish everything is fine. Now we'll continue in lecture number 7, and this is one of the most important lecture in this module. It's how to interpret your multiple sequence alignment, and this is very important to understand and to know how to do this. So simply the interpretation of your multiple sequence alignment, it depends on how much its appearance. So you have to look first at the appearance of your multiple sequence alignment, then you can try to depend on it to interpret your result. Some tools on the internet can help you make sense of your various alignment by extracting first. You have to extract some blocks that is very useful or looks useful, and then you try to single out special position. Like you have to find special positions that make sense, and then try to single them out. So this will help you in your interpretation. So these two things it will depend. You have to depend on them to interpret your result. Okay, interpretation, as we always say, in alignment is a bit of art, not just science. It's a bit of art, but it's actually this art is based on science. So that's why you have to use that carefully. E-values, what the second thing is very important, and the E-value means, for the people who don't know, the score that tell you how reliable your database search is. So this is very important thing that you have to be aware of. So that means that deciding whether your alignment is correct still involves some educated guesswork. So you need to understand this I mean to be able to continue your research. So we have to know that the DNA alignment are by far the most difficult to interpret. So that's why DNA is much more harder and more difficult than the protein. Interpretation of protein DNA alignment is much easier than DNA alignment. If you analyzing this type of sequence, you want a very high level of conservation, knowing that a single conserved column are likely to be meaningless. So for the DNA, single columns of, as we said, single columns is meaningless, is not enough. So, but in protein conservation, for few as we will see now, will have a meaning. If you have a protein sequences and there is some column are conserved, this will have a meaning. And we will tell you about this later and how we can compare and what is the threshold for each one of them. A DNA block is only informative when it contains several identical columns in a cluster. So for DNA, you have to have several or identical columns. But even with the DNA of closely related sequences, obtaining such an alignment is still difficult. Okay, this is why most biologists prefer protein alignment because it doesn't need a lot of DNA blocks to just be convinced that you have a good alignment and have a biological meaning. So that's why we prefer using protein. Recognizing the good part, of course, in protein alignment, the most convincing evaluating grid we have for a protein multiple sequence alignment stem from our knowledge of protein structure. So based on our knowledge of protein structure, we have to understand what is the convincing grid of that tell us what is the best part of the multiple sequence alignment we can see. Of course, we have to just quickly, we know that the protein, we have the primary protein structure, which is a sequence of a chain of amino acids. We have also the secondary structure of protein, which give us the alpha helix and beta pleated sheet. And then we have the tertiary structure, when occurs when certain attraction are present between alpha helix and beta plate sheets like here, and we have also the quaternary structure, protein structure, and this happen when a protein consisting of more than one amino acid chain. So this is based on our knowledge, the primary, secondary, tertiary, and quaternary structure. Based on that, we can try to interpret our multiple sequence because we will look for amino acids columns that are conserved and have relation with this structure. So we know that the structure contains surface loops that evolve rapidly, like this ones here, this surface loops. And these loops are softer portion of the protein that connect its more rigid portion. The more rigid portion he here means the alpha helix and beta sheet, which is like this one here, this one. So this alpha helix and beta sheet. So when you look here at arrow number 1, we are looking for the places that have making the strand and making helixes. So we have here to see where is the amino acid that are conserved and making straight and other places that making helixes. So here we have helix, helix, and here this amino acid conserved position for making strand and making strand here and strand here. So this can make us understand why the structure of these sequences are conserved. So this because this important structure of the rigid portion of the protein are conserved. So this give us this indication in our interpretation. But the other places, or the like here, this place and this place and here. So this is the one that have a lot of mutation. Usually this is the places with where it makes the structure of the surface loops. So that's why it will you will find a lot of mutation in it, like here, like here, like here, for example, and this loop, and that's it. So that's why you have to understand this before you continue your interpretation. Protein structure also contain core region that act as support walls for the protein. These support walls evolve less rapidly, as we said, than loops, of course. So the places that make our helixes and alpha helix and beta sheet evolve less rapidly than the one that makes loops, as we said before. Okay. So in the new multiple sequence alignment, you can expect to find, of course, nice gap-free blocks that correspond to the core region, which is alpha helix and beta sheet, and gap-rich region that correspond to the loops, as we can see here. Here is places for the helixes, places for the sheet. Here, this position, you will find the gap-free region here in the helix and sheet. But the position that contains a gap-rich region, this corresponds to the loops. Okay. Also, you have to understand in interpreting there is many what you call capitalistic signs. The last line contains seemingly cluster W, of course, of in the or muscle or tree coffee alignment, what we call capitalistic signs. What are these capitalistic signs? These capitalistic signs like the star and the colon and the period. These three signs are very important. What the meaning of the star? Star indicate an entirely conserved column. So if you have a conserved column in your multiple sequence alignment, you will find this star under it, which means that this column is entirely conserved. If you find a colon under your multiple sequence alignment, this indicates that this column where all the residue have roughly the same size, have roughly the same size and the same hydropathy. So this is not identical, but similar. So similar amino acids, you will have this column. But the column which has identical amino acid, we will have star under it. So if you see a period under your column, this means that there is size or the hydrophobic. hydrophobicity of this amino acid in that column is similar. So and preserve it in the course of evolution. So these three capitalistic signs are very important. So the first is the star, which indicate the identity, the amino acid in that column are identical. And we have similarity based on hydrophobicity or same size. If you see that sign, this means that the amino acids in this column have the same size and hydrophobicity, both of them. But if you see only period under this column, this means that this column contain amino acids that may have the same size or hydrophobicity or has the same preserved in the course of evolution. Okay. If you take a look at this block of amino acids, for example, the average good block is a unit of at least from 10 to 30 amino acid long. So you have something like this. This exhibit at least one of two, three stars, like here, one, two stars, and maybe more, a few more colons close to the stars, and several periods scattered along the MSA result. Something like this, and maybe more or less, but that's what we call that's average good block. So the good block have at least from 10 to 30 amino acid long, exhibiting at least one or two, three stars, which mean identical, few more colon like this ones, and this means that it's conserved but not identical, and have the same hydrophobicity and size, and several periods, which mean this column has amino acid in that column have either the same hydrophobicity or the same size. So this is one of the good things that you can get. The magic thing about multiple sequence alignment is that 4 or 5 conserved position over 50 amino acid can be enough to convince us that we are looking at a genuine signal. This is less than 10% actually, but this is a good reason to understand that these symbols are very important to us to understand and to interpret our result. You have to remember that we require at least 25% identity to consider a pairwise alignment. So if you would like to consider that there is a pairwise alignment between two sequences and there is common ancestor or something, you have to understand that you at least need 25% identity. And this is very important, at least. Not similarity, but identity. So conserved column in a multiple sequence alignment are meaningful only when the surrounding columns is not conserved. This is one of the things you have to put in mind also. And another criterion for a useful multiple sequence alignment is knowing that the type of amino acids you can expect to see conserved. It's very useful to understand what type of amino acids are conserved. This is also very important, and we will tell you why now. Amino acids aren't equal, and they all have very characteristic pattern of mutation, conservation, and a multiple sequence alignment. And we will see as we see. There is pattern of conservation in multiple sequence alignment we're looking for. So if you see tryptophan or phenylalanine or tyrosine, this is if you see that kind of amino acid are exist and conserved. In this case, you have to interpret it in a very certain way because it's common to find conserved tryptophan as a large hydrophobic, as we said, a residue that it's deep in the core of the protein. So that's why if we see a conserved tryptophan, it's an important role in their stability and therefore difficult to mutate. So when tryptophan mutate, it's usually replaced by another aromatic amino acid, such as phenylalanine or tyrosine. So that's why we are looking for that conserved columns, that if it has tryptophan or phenylalanine or tyrosine. And this pattern of conserved aromatic amino acid constitute the most common signature for recognizing protein domains. So that's what we're looking for. That if you're looking for protein domains, so you would like to look for conserved columns of tryptophan or phenylalanine or tyrosine. Okay. Next is glycine or proline. Glycine and proline is common to find conserved columns with glycine or proline in the multiple sequence alignment. These two amino acids often coincide with the extremities of well-structured beta strand or alpha helix. So if you are looking for to see the well-structured beta strand and alpha helix, then you are looking for glycine or protein conserved columns. That is and to find, so that's a good indication to understand and to interpret your conservation of that column. Okay, now we'll talk about cysteine. What about if I see if I see for example column that with conserved cysteine like this one here, this one here, this one here, this conserved positions of cysteine only amino acids. So take a look at here. Cysteine are famous for making cysteine disulfide bridges, disulfide bridges. Conserved column of cysteine are rather common and usually indicate that such bridges like this bridges here and such bridges here and column of conserved cysteine with specific distance provide a useful signature for recognizing domain and faults as we see from this figure here. So this is one of the things we are also looking for. So if you are looking for this kind of research for that kind of information, so you are looking for the cysteine columns and where its position are conserved as we see here. What about histidine and serine? Histidine and serine are often involved in catalytic sites. So the people who are studying catalytic sites of protein like here or here, so in this case, you are especially looking for this column that have a conserved histidine or serine. For example, especially those of proteases and a conserved histidine or a conserved serine is a good candidate for being part of the active site. So here is active site here or active site here. So that's why we are looking for histidine like this one, serine 195 or this histidine 57. This should be conserved among different species, or you can see that conserved in the multiple sequence alignment because it usually, as we said, it's a part of the active site. Okay, what about other amino acids like lysine, arginine, aspartic acid, and glutamic acid? These charged amino acids are often involved in ligand binding, and that's very important too. And there is many people who are looking for this sites in your protein, the ligand binding site. So highly conserved columns can also indicate salt bridge inside the core of the protein. So if you are looking for binding, ligand binding like here, these places usually you will find like lysine or arginine, aspartic acid, or glutamic acid. Also, as we said, it's also can be inside the core. Salt bridge indicate a salt bridge inside the core of the protein also. So this is very important. Okay. Now we have also lysine, leucine. Leucine are very rarely conserved, and unless they are involved in protein-protein zipper, such as in this kind of interaction. So as you see, leucine are very rarely you can find it conserved, but as we see, if this a place, if you have this information about your protein, about leucine zipper, so its interaction like protein-protein zipper, as we see, you can find this amino acid are conserved. So, okay, so this is many things. It's very important to know the interpretation of your conserved amino acid columns and why it's conserved and how we can interpret it using this information. So, and of course, whatever you are looking for, there is many things you can look for, and based on that, you can interpret your multiple sequence alignment. I hope you can try to practice your multiple sequence alignment and find this conserved position and try to understand why it's conserved and what's its important based on this lecture. Thank you very much, and see you in our next lecture.